Hello, welcome to Biology, the scientific study of life. Molecular clocks help track evolutionary time. As we stated at the beginning of this chapter, one of the goals of evolutionary biology is to understand the relationships between all living organisms, including those for which there are no fossils. When we extend molecular phylogenes beyond the fossil record, however, we must rely on an important assumption about how change occurs at the molecular level. Molecular clocks. Recall our statements earlier that researchers have estimated that the common ancestor of Hawaiian silverswort lived about 5 million years ago. How did they make this estimate? It relies on the concept of a molecular clock, a yardstick, for measuring the absolute time of evolutionary change based on the observation that some genes and other regions of genomes appear to evolve at constant rates. The assumption underlying the molecular clock is that the number of nucleotide substitutions in orthologous genes is proportional to the time that has elapsed since the species branched from their common ancestor. In the case of paralogous genes, the number of substitutions is proportional to the time since the genes became duplicated. We can calibrate the molecular clock to a gene that has a reliable average rate of evolution by graphing the number of nucleotide differences against the times of a series of evolutionary branch points that are known from the fossil record. The graph line representing the evolution rate of this molecular clock can then be used to estimate the date or the date of evolutionary episodes that cannot be discerned from the fossil record, such as the origin of the silverswort. Of course, no genes mark time with complete accuracy. In fact, some portions of the genome appear to have evolved in irregular fits and starts that are not at all clock-like. And even those genes that seem to have reliable molecular clocks are accurate only in the statistical sense of showing a fairly smooth average rate of change. Over time, they may still be chance deviation above and below that average rate. Finally, even among genes that are clock-like, the rate of the clock may vary greatly from one gene to another. Some genes evolve 1,000,000 times faster than others. Neutral theory. The regularity of change that enables us to use some genes as molecular clocks raises the possibility that many of the changes in these sequences result from genetic drift, and the changes are mostly neutral, neither adaptive nor detrimental. In the 1960 years, Jack King and Thomas Jocks at the University of California, Berkeley, and Mato Komura of the Japanese National Institute of Genetics published a paper supporting this neutral theory, that is, that much evolutionary change in genes and proteins has no effect on fitness and therefore is not influenced by Darwinian selection. Komura pointed out that many new mutations are harmful and are removed quickly, but if most of the rest are neutral and have little or no effect on fitness, then the rate of molecular change should indeed be regular like a clock. Differences in the rate of the clock in different genes are a function of how important each gene is. If the exact sequence of amino acids a gene specifies is essential to survival, most of the mutational changes will be harmful and only a few will be neutral. As a result, such genes change only slowly, but if the exact sequence of amino acids is less critical, fewer of the new mutations will be harmful and more will be neutral. Such genes change more quickly. Difficulties with molecular clocks. In fact, the molecular clock does not run as smoothly as neutral theory predicts. Many irregularities are likely to be the result of natural selection in which some DNA changes are favored over others. Consequently, some scientists question the utility of molecular clocks for timing evolution. Their skepticism is part of a broader debate about the extent to which neutral genetic variation can account for DNA diversity. Indeed, new evidence suggests that almost half of the amino acid differences in proteins of two Drosophila species, D. Simulans and D. Yacoyoba, are not neutral but have resulted from directional natural selection. Nonetheless, it is likely that other extremely long periods of time fluctuations in evolutionary rate due to selective pressure will average out, so that even genes with irregular clocks can serve as approximate markers of a ellipse at that time. Another question arises when researchers attempt to extend molecular clocks beyond the time span documented by the fossil record. Although some fossils date back as far as 3 billion years, this year very rare and abundant fossil record extends back only about 550 million years. But molecular clocks have been used to date evolutionary divergences that occurred a billion or more years ago. These estimates assume that the clocks have been constant for all that time, though such estimates are likely to have a high degree of uncertainty. Applying a molecular clock, the origin of HIV. Recently, researchers at Los Alamos National Laboratory in New Mexico used a molecular clock to date the origin of HIV infection in humans. Phylogenetic analysis shows that HIV virus that causes AIDS is descended from viruses that infect chimpanzees and other primates. The viruses do not cause any AIDS-like disease in non-human hosts. When did HIV jump to humans? There is no simple answer, because the virus has spread to humans more than once. The multiple origins of HIV are reflected in the variety of strains, genetic types of the virus. HIV's genetic material is made of RNA, and like other RNA viruses, it evolves quickly. The most widespread strain in humans is HIV-1M. To pinpoint the earliest HIV-1M infection, the researchers compared samples of the virus from various times during the epidemic, including one partial viral sequence from 1959. The samples showed that the virus has evolved in a remarkable clock-like fashion since 1959 by extrapolating from their molecular clock. The researchers concluded that the first HIV-1M invasion into humans took place during the 1930s. The universal tree of life. In the 1960s, researchers deciphering the genetic code found that it is universal in all forms of life. They inferred that all present-day organisms must therefore have a common ancestor. Today, researchers are applying systematics to link all organisms in a tree of life. Investigators use two criteria to identify regions of DNA molecules that can demonstrate the branching pattern of this tree. The regions must be able to be sequenced, and they must have evolved so slowly that homologies between even distinctly related organisms can still be detected. The rRNA genes, which code for the RNA parts of ribosomes, fit these requirements because rRNA genes are fundamental to the workings of the cell. Their molecular clock runs so slowly that they can serve as the basis for a universal tree of life. But for the moment, let us make 2 points. One, the tree of life consists of 3 great domains: bacteria, archaea, and eukarya. The domain bacteria includes most of the currently known prokaryotes, including the bacteria closely related to chloroplasts and mitochondria. The second domain, archaea, consists of a diverse group of prokaryotic organisms that inhabit a wide variety of environments. Some archaea can use hydrogen as an energy source, and some were the chief source of the deposits of natural gas as that are found throughout Earth's crust. The third domain, eukarya, consists of all the organisms that have cells containing true nuclei. This domain includes many groups of single-celled organisms, as well as multicellular plants, fungi, and animals. 2. The early history of these domains is not yet clear. Comparisons of complete genomes from the 3 domains show that, especially during the early history of life, there have been substantial interchanges of genes between organisms in the different domains. These took place through horizontal gene transfer, in which genes are transferred from one genome to another through mechanisms such as transposable elements, and perhaps through fusions of different organisms. The first eukaryote may have arisen through such a fusion between an ancestral bacterium and ancestral archaean, because phylogenetic trees are based on the assumption that genes are passed vertically from one generation to the next. The occurrence of such horizontal events means that universal trees built from different genes often give inconsistent results, particularly near to the root of the tree. As a result, details of the tree are continually being revised. However, the division into 3 great domains has remained secure since systematists first glimpsed the overall shape of the universal tree.